this lecture, we will be discussing about an introduction or the basics of how we do models or how do we model biological systems. So what is, in this discussion, we will first briefly define modeling and what is the modeling process. We will also define the scales and complexity of some models. And then what are the difference between the deterministic models and the stochastic models. And we will also finally discuss the different approaches to modeling, which is the top-down and the bottom-up approaches. So first, what is modeling? Modeling is a human activity consisting of representing, manipulating, and communicating real-world daily life objects. We basically have here, we need to define or model a system. A system which is a collection of interrelated components or interrelated objects. So there are three primary scientific uses of models. So for instance, if you have a certain system which we did not know much about, so it's under a specific external stimulus E, then it also produces a certain response R. So you want to know the impact of the stimulus to the response. Now, the first use of modeling to know that is to synthesize or knowledge discovery. So to use the knowledge of the inputs E and the outputs R to infer the characteristics of a system. For example, your system is a certain enzyme system. So your enzyme catalyzes the production of a product from the substrate. And your input is, say, an inhibitor, a molecule. So an external molecule, your external stimulus, you place it on your system, and then you observe that the activity of the enzyme somehow decreases. So you can see now that substance your input E, is an inhibitor or may act as an inhibitor of your system. So using another model, you can also analyze and predict the behavior of the system. So you use the knowledge of the parts of the stimulus to account for the observed response, and eventually to predict the response to different stimuli. So for example, if you have your, again, the enzyme substrate system, you input the inhibitor. So varying the components of the inhibitor, the concentration of the inhibitor in relation to the substrate, you manage to actually infer that the inhibitor you have is the type of a competitive inhibitor to your enzyme. So the third use is instrumentation or device. It's used to design an alternative system, so to produce the input-output relationship with the best possible adherence to the studied system. For example, that enzymatic reaction is actually important in the process of inflammation. So you can design or you can further develop using the idea from the substrate inhibition. You can develop an inhibitor or a blocker for that certain metabolic pathway in such a way that you can actually develop, say, anti-inflammatory drug if that enzyme has something to do with the process of inflammation, something like that. So other uses of models, so to account for conceptual frameworks to design new experiments. It's also a method to summarize or synthesize large quantities of data. For example, in bioinformatics, you have a very, very large amount of databases. You have many interacting genes, for instance, genes and proteins. So you can use modeling in order to make sense of those large amounts of data, because otherwise it's just data without anything. You cannot, it's difficult, without modeling, it's difficult to extract information, raw information from data. So tools to discover relationship among objects, so like that one. So you can infer this is an inhibitor, or this is an activator, or this is the increasing this amount causes another increase. So correlations, causalities, you can also do that in modeling. So you have your modeling process here. So for example, you have your real world problem or situation. You formulate a problem from an observed problem, or you can say an observed situation in real life. So you simplify it in order to properly formulate your problem, but of course you still need to validate that. And then if you formulated your problem, you do mathematization, or you try to create mathematical models or equations or graphs. So try to describe your problem. So that using that mathematical models, you find the solution within your models and interpret that solution. Does your solution is that solution answers the problem you formulate? And if so, does it validate the real world situation? Does it apply to the real world situation you have? Something like that. So the modeling process, we have 3 basic steps. So first, the model implementation consists of describing a formal language, the objects, relationships identified in the system under study using mathematical structure or a computer code. So you implement first the model. Afterwards, you use that model to forecast the system behavior. And finally, you need to check if your model is actually a good representation of your system. So you need to evaluate the adherence of the model to reality by matching the conditions from experimental data. Now, here is an important thing about modeling. It must not be fully removed from experimental data. You still need to validate the authenticity or the plausibility of your model using actual experimental data, because regardless of that, it will just be a simple dream without actual experimentation. So here is some more detailed steps in modeling. At the end of this course, if I remember correctly, I'm asking you to create a simple model of a certain biological phenomena. So it will do you well if you will remember this in doing your paper. So if you have, at the start, you observe a biological phenomenon, and then you first formulate what is our objective, what do I want to do with my model. For example, I want to model the binding, the cooperative binding of, say, hemoglobin, or rather oxygen to hemoglobin. So if that is my objective, I want to create a model on that. So you need to know as much as you can, as many as you can, the possible knowledge you have. So it's a very extensive literature search. So you need to know all of the background knowledge published on that, so that your model can be as close to real life as possible. And then you create the model. So your model structure, you structure the model, you create a hypothesis for that, and then create a conceptual model, and then formulate it mathematically. So after formulating it mathematically, you can use a computer to easily solve your model, to create graphs, to find solutions for these mathematical formulations. Then you calibrate your model. So in calibration, you need to check the parameters if it is as close to reality as possible. So it's more of sometimes you have your values from the current literature. Sometimes you need to do some tweaking, that is trial and errors. So you create trial runs after parameter estimations. You create trial runs against a model construction data set, and then you compare the model output to the test data set. So does it follow? Are your objectives satisfied? Do your models reflect reality? So if it's yes, so that's good. So your job is done. But it's not, you go back to the formulation, you recreate the tweaks. So you redo your models, you redo your calculations, you redo some equations. You might need to add some other parameters, or you might decrease some other parameters. You simplify things, or you might need to increase the terms. And then again, you do solutions, you do testing, you do running, and then you double check, does it still compare? But still not. So you have your, again, mathematical formulation. So if you do not think there's anything wrong with your math formulation, you can check if your hypothesis is wrong for the objectives. So for example, you want to see the cooperative binding, but you're actually testing it on an inhibitor, so it might not be correct. So you do evaluate the objectives of your study. So until you create a good, or such that the objectives are satisfied. Okay, so next we will be discussing scales of a model. So in choosing a scale, it depends on which aspect of the phenomena, from the micro scale to the macro scale, you are interested in analyzing. So the scale is the basic unit available for defining a scale is the cell with no regard to its physical dimension. So you can also use subcellular scale, intracellular scale between cells. You have the cellular, mesoscopic or intercellular scale, which is the macroscopic scale, or beyond that we can do the population dynamics or population scale, so a scale of a whole population or community of actual organisms. So the complexity and multi-scale model. So a complex system is a system composed of different interconnected parts such that, as a whole, they exhibit one or more properties which do not obviously arise from the properties of the individual parts. And that is, most of them, you can see them in biological systems. The biological systems are mostly complex. So there are two types of complexity. We have disorganized complexity, which is the complexity arising from a very large number of parts. And we have the organized complexity. The complexity is intrinsic to the system, and eventually with the limited number of parts and its connection rules. So we have those types of systems. So a good example of a system which is quite we have this organized complexity here. Say the immune response. There are lots of many different components. When we say immune system, it's not just about the white blood cells. In fact, in the white blood cell, it's just a category of cells. There are lots of white blood cells. And then you have the organized complexity, which even with the limited number of parts, there are still some rules that needed to be considered, which gives it complexity here. Now, we have two types of models that define our systems. So we have the deterministic models and we have the stochastic models. So what are the two? So deterministic models are usually solved through calculus-based methods. So the outcomes are precisely determined through knowledge of the relationships between variables. So when you do the solutions, you always end up with a small range of solution. There's always this one correct answer. So you provide a dynamic and quantitative description of a spatially homogeneous system. So one thing about deterministic models is that for it to be possible, it must be as simple, your system must be defined as simple as possible. So you have it as homogeneous as you can. So it usually defines system as perfect and without noise. So sometimes the downside of that is that it can be oversimplified. So example of that, the law of mass actions and rate kinetics are usually deterministic models. If you know the substrate, the amount of substrate, you can determine the rate of the reaction. So those are types of deterministic models. In contrast, you have stochastic models. Stochastic models are mathematical models for non-deterministic physical systems. So stochastic is more of probabilistic. So it can adapt many possible behaviors starting from any given initial conditions. So as I said, it's probabilistic. So you only end up with, say, for example, this is a 90% chance of going this way, but you still end up with another 10% chance of going another way. So something like that. So it possesses inherent randomness and accounts for the noise in the system. So in this system, you can input the noise, you can consider the noise in your system. So it's more, unfortunately for us, although if we consider biological systems, stochastic models are really a good way to model them because biological systems are quite random, they are very complex, and they have a lot of noise. However, the problem with this type of model is that they are very complex and difficult to analyze. That means you need to expend more computational effort just to solve them. Sometimes it's very, very difficult to just run a very simple model, unlike when we run the deterministic counterpart of that model is quite fast, and then we do the stochastic counterpart. It's very long. It's more complicated. It requires a lot of computer time. So the next one is about top-down and bottom-up approaches. So in modeling, you consider two approaches on how you approach your system. The first one is the bottom-up approach. You started with the smallest possible component, for example, a simple enzyme reactions, and then you go up. So this is usually employed in systems biology, where you want to model systems as part of you want to create models as part of a whole system. So you perhaps start with a certain metabolic reaction, a certain let's say glycolysis. So start with that, and then you move up. So you input how does glycolysis interact with other metabolic reactions. For example, the urea cycle. How does it connect to the urea cycle? How does it connect to glyconeogenesis, for example? How does it relate? How does it relates? How does it connect to the synthesis of some amino acids? Something like that. So you start with small and you proceed upwards, and then eventually, for example, how does it connect to other? Going back to our example on glycolysis, you know how to you model, or you approach it on glycolysis, and then how it interacts with the other metabolic reactions, with the metabolic pathways, and then going up, how does that as a whole affect the system, or how does it as a whole affect your organism. Whereas in the top-down approach, it's a more of a bioinformatics way. So you start with the large data set. So usually it's an mRNA, or you have RNA sequences, you have your microarray data or RNA-seq data, and this is a whole holistic type of data. You're actually looking at the data from the whole cell, and then you do bioinformatics approaches. Also, some those are applying graph theory. It can be knowing the calculating statistical calculations on what are the over and under expressions, and from that you want to understand the process at a more basic level. So for example, you have a microarray data and you want to model a metabolic system from that. So that is the top-down approach. So another view of that, so you have here in a bottom up, you start with small. It can be a certain simple theory, say this enzyme is this substance is an inhibitor of that enzyme, and then you go up. So you look at reconstruction of the network, how it relates to the organism, or you start with the actual data and you try to create a theory from that. So those are the two approaches, the top down and the bottom approach. So these are the references if you want to have further readings on that, and we will see you again next time.